Welcome to this explainer. Today, we're diving straight into the brilliant mechanical and electronic engineering that actually makes human health in space possible. We're doing a full teardown of this fascinating piece of tech called the SAP-1 Payload. Basically, we're looking at how engineers are building a fully autonomous, completely miniaturized laboratory specifically designed to run complex biological experiments in the extreme, harsh environment of low Earth orbit. It's an absolute masterclass in solving massive problems inside microscopic spaces.
So, right off the bat, we're hit with this huge, mind-bending question. How on Earth, or rather off Earth, do you grow a trillion bacteria in zero gravity? Space is just incredibly hostile. You've got no natural gravity to separate your liquids and gases. And without gravity, you actually lose natural heat convection. So hot air literally doesn't rise, liquids don't settle. This is a monumental hurdle when we need to study essential biology, like our gut microbiome, to keep astronauts healthy on those long missions. Everything we take for granted in a standard lab down here on Earth just stops working.
Okay, here's our blueprint for today's mission. We'll start with Section 1, the Space Microbiome Challenge. Then move to Section 2, inside the SAP-1 Payload. After that, Section 3, the Rotating Platform Solution. Section 4, the Electronic Brains. And finally, Section 5, the Future of Space Health.
All right, diving into Section 1, the Space Microbiome Challenge. Let's really look at the biological why behind this incredible piece of space hardware. I mean, why are we even sending bacteria up to space in the first place?
Well, the effects of spaceflight on the human body are just profound. Astronauts deal with severe dysregulation in their gastrointestinal tracts, and that's thanks to microgravity, high stress, and radiation. But, this isn't just about upset stomachs or digestion. Recent research actually confirms that our gut microbes are heavily tied to our immune system, and crucially, the gut-brain axis. Shifts in these gut bacteria are directly linked to cognitive status and mood. So if we actually want humans to survive long-duration missions to, say, Mars, fixing this microbial imbalance in space isn't just a nice-to-have. It's absolutely a top priority.
To tackle these health issues, scientists are zeroing in on this tough little probiotic called Bacillus clausii. The secret sauce here is that it's spore-forming. This bacterium has honestly a survival superpower. It can form a really hard protective shell and enter a dormant state as a dry spore, which means it can safely ride out the extreme, violent conditions of a rocket launch without dying. Once it finally reaches a hospitable environment, like a human gut, or in our case, the SAP-1 payload, it germinates, wakes up, and starts growing.
Moving on to Section 2, inside the SAP-1 Payload. Now that we know exactly who our microscopic passenger is, let's zoom in and check out the physical anatomy of the actual spacecraft payload designed to house these bacteria up in orbit.
The engineering constraints for this mission are absolutely extreme. The entire module has to run on a peak power of barely 5 watts. I mean, that's literally a fraction of what a standard LED light bulb uses. And on top of that, it has to weigh under 1,500 grams, and the whole shebang has to fit inside a tiny 10-centimeter cube. In aerospace terms, that's called a 1U form factor. We are talking about squeezing an entire microbiology lab into something the size of a Rubik's Cube.
To actually pull this off, the SAP-1 features a hermetically sealed outer shell. Because the vacuum of space would instantly boil off any exposed liquids, this chamber is filled with nitrogen gas and kept at exactly 1 bar of pressure. That perfectly mimics Earth's atmosphere at sea level. This sealed environment acts as the foundational, protective bubble for everything going on inside.
Now, tucked inside that chamber are these tiny, transparent biowells where the bacteria actually live, but you can't just fill them to the brim with liquid. Extensive ground tests revealed a very precise sweet spot, exactly 75% air to 25% culture liquid. Why? Well, because in zero gravity, fluid dynamics go completely out the window. Without gravity to pull the liquid down, the bacteria absolutely need this specific pocket of air to guarantee they get optimal aeration and oxygen. Otherwise, they just suffocate in their own nutrient broth.
Which brings us right to Section 3, the Rotating Platform Solution. The big crucial question here is, how do we actively feed and measure these bacteria without gravity mixing things up for us?
And that brings us to the core mechanical innovation of SAP-1. It relies on a rotating platform mounted directly on a stepper motor sitting dead center in that sealed chamber. The biowells are placed on the outer edges, while a pressurized nutrient tank sits right in the middle. You can think of it as this micro-scale carnival ride for our bacterial samples. and it brilliantly solves multiple zero-gravity problems all at once.
So, when it's finally time to wake the bacteria up from that dormant spore state, the payload runs through a rhythmic, hyper-precise mechanical sequence. Step 1, tiny solenoid valves snap open. Step 2, that pressurized central tank acts kind of like a syringe, pushing the liquid nutrient broth through these tiny pipes into the biowells on the edges. And then, step 3, the platform executes a rapid, jittery sloshing motion. This physical agitation perfectly mixes the dormant spores with their food, making sure they don't just clump together in a big floating bubble.
Once they're actively growing, we obviously need to measure them using lasers. But because liquids and air bubbles just float around randomly in zero-G, you can't get a clean optical read. The laser would just bounce off a random air bubble. So they use what's called the centrifuge analogy. By spinning the platform at a steady 5 revolutions per minute, it creates just enough centrifugal force to push the heavier liquid culture to the very far end of the biowell. The air stays near the top, the liquid pools at the bottom, and bam, the optical sensors can shoot a laser cleanly right through the liquid to measure the bacterial growth.
It really is this beautifully elegant, all-in-one solution to some incredibly complex orbital problems. The motion isn't just for separating the fluids either. That jittery mixing prevents the bacteria from clumping up and sticking to the glass. The rotation itself actually acts as a passive backup pump, pulling liquid outward if the pressure system ever fails. And critically, remember how hot air doesn't rise in space? Well, physically spinning the platform churns the nitrogen gas inside the chamber, forcing mechanical heat convection so that the bacteria stay nice and uniformly warm.
Okay, let's jump to section 4, the electronic brains. We need to dig into the electronics to see how this intricate mechanical dance is actually controlled, all while sipping less power than your smartphone charger. This detailed blueprint reveals the true nervous system of SAP-1. Everything we just talked about, the motors, the heaters, the sensors, it's all routed through an onboard command and data handling unit. And since space radiation can totally scramble normal computer chips, this system uses flight-tested microsemi Smart Fusion 2 processors. It coordinates the timing, stores the data, and talks to the satellite's main communication bus using a specialized slip ring. That's a crucial little component that lets the platform spin endlessly without tangling up any of the wires.
Yeah, remember that incredibly strict 5-watt power limit? The engineers had to get really creative here. To save precious power, the system uses a super clever spike and hold circuit for the solenoid valves. Opening a valve against pressure takes a ton of energy, so the system blasts a quick 5-volt spike to snap the valve open instantly. But once it's open, it takes just a fraction of that energy to keep it there. So the system immediately drops the voltage down to 3.3 volts to hold it open, which saves massive amounts of power over the entire mission.
And to actually measure how fast these bacteria are reproducing, the engineers pulled off another incredible shrinking act. They took a massive, heavy, benchtop lab spectrophotometer and literally miniaturized it into a tiny digital light sensor. It's tuned exactly to a 600 nanometer wavelength. At this specific optical density, the light isn't absorbed by the clear broth, but it is blocked by the solid mass of the multiplying bacteria. So simply put, the less light that hits the sensor, the more bacteria have grown.
Finally, we've arrived at section 5, the future of space health. Now that we've completely torn down the hardware and the electronics, let's zoom out and look at the bigger picture of how this mission will actually unfold in orbit.
The payload is designed to operate on a highly controlled thermal timeline. During launch and storage, it keeps the dormant spores at a stable 30 degrees Celsius. When the experiment starts and the food is released, it carefully heats the biowells to 35 degrees to perfectly simulate the internal temperature of the human body. But arguably the most critical part is phase 3, a 120 degrees Celsius kill switch at the very end. The heaters will just crank up, totally sterilizing everything inside to ensure absolute biological containment in space. Trust me, we do not want to accidentally seed Earth orbit with mutated space bacteria.
As SAP-1 paves the way for astronaut gut health using this brilliant microengineering, solving the complex problems of zero gravity with rotating platforms and laser sensors, it leaves us with a profound final thought. What other natural biological phenomena, things we take entirely for granted down here on Earth, are we going to have to completely hack and re-engineer from scratch to survive in the void of space? This payload is just the very beginning of humanity learning to take our biology out into the cosmos. Thank you so much for joining me for this explainer, and keep questioning the universe around you.